Morning, everybody. It's a gorgeous day today, and spring is around the corner. I noticed hyacinth plants popping out in our garden, and I'm sure that's happening around the whole Fayetteville, Bentonville, Northwest Arkansas region. Is the daffodils and all of those summer, winter drops or wherever those things come out, the white little bell-like flowers that they're probably all popping out. So my name is Ranu Jung. Someone from the audience once said, I really don't know who you are. One of our visitors. So I'm here at the University of Arkansas. I'm the founding executive director for the Institute for Integrative and Innovative Research. I'm also a professor of biomedical engineering, and you'll see the link there in a minute, and associate vice chancellor. So as many of you are here and hear this every time I talk about it, but every time we also have members on Zoom, and we don't know who all they are, and every time there are new people who join. So I want to start the day actually today with a really strong, really exciting news. And that exciting news is that Meredith Hadkins, who is in our audience, our IQBR assistant research professor, Philip Sample, and a whole other team, won phase 2 of the NSF Convergence Accelerator. Some of you who were paying attention last year, remember we had that Convergence Accelerator panel, and we have talked about sustainable proteins, we have talked about food and connecting food, access of food to the community and all of that different parts, and bringing technology in here. So you'll get to hear more about it later, and watch out for the new big press release that'll come out broadly on Monday. But the university got news of it today, and so this is really, really exciting. Very hard one, a very big collaborative project, not just within the university, multiple universities pulled together, and industry partners pulled together. So great way to start your morning when you open the Arkansas News and see that kind of a affirming article there. IQDAR, as you know, we are committed to convergence research, solving wicked problems, and finding solutions that bring in integration and innovation, but the solutions that can be scaled with a very core mission to make sure that we have societal impact. So in this, we have said one of the first big grand challenges we will take on, and the problem that we take on is integrative health. And when we say integrative health, we are saying it has everything to do with our well-being, but also the society around us, of how we are going to create situations that will things well. One of the things that happens with integrative health is how much you sleep. And because sleep impacts a lot of different things of our body. And then as you change across the lifespans, things happen differently. One thing that gets really impacted by sleep also is memory. And there are short-term consequences and long-term consequences of it, of what happens with the memory itself. So today we actually have somebody who is here with us, a leader in this technical field, and a very well-known a researcher who's been combining computational work and experimental work, converging two different worlds together, but not just of neuroscience, but also of biomedical engineering and mathematics, to understand the basis. What is the basis of the neuronal basis for memory? I'm not gonna give this talk. He's gonna tell the talk later. But I am so excited to welcome my old friend, John White, who I think is a friend to many, many people in the community, because he was also the president of the Biomedical Engineering Society. You know what? That is our for the field. That is the premier the society for us. And so John is a friend to many, and he's a friend to many, not just because he's a leader, but because of his personality, his nature, and his willingness to embrace all students to all the rest. He's the chair of the Biomedical Engineering Department at Boston University that has arguably really risen very fast to the top. There's multiple faculty, so we'll be looking to get your guidance as we build IQSTAR. But he's authored over 100 publications, many invited lectures. He's a fellow of the Biomedical Engineering Society, the American Institute for Medical and Biological Engineering, and as well as the International Academy of Medical and Biological Engineering. Also, for the students, when you get a chance to talk to him, he has a long-standing National Institutes of Health training grant on quantitative biology and physiology. So pick his brain about what it means to be, going through a training program and the future for that. So, John, welcome to Fayetteville, welcome to University of Arkansas, welcome to IQSTAR, and welcome to our seminar series.
Thank you. Thank you, Ranu. Ranu is an old friend of mine, and it's just, I'm delighted that she's here. I've been to Fayetteville a few times in my life, for family reasons. I have family nearby. And, but this is really my first time visiting this university as a biomedical engineer. And it's just amazing what has been accomplished here. I mean, the department is about 10 years old, and it's just doing great. And this is, an effort that is very exciting. I mean, we have this tug of war and hopefully communal effort that we are, as scientists, engineers, and physicians, work together to sort of build our knowledge base and then figure out how to use that knowledge appropriately to improve human health, which is the only reason, which is the main reason that we're in this job, right? It's certainly the reason that the National Institutes of Health might be willing to fund us. And I think that I'm very excited by this idea that Ranu and the rest of you are launching, this idea that we're going to speed up the translation part. We're going to basically try to have impact in the earlier date. And I'm gonna, my work I'm gonna talk about is, a lot of it is discovery, but I'm gonna end with a few words about how one might build memory prosthesis. And I'm always worried. I just met a psychologist. I'm always worried that there are psychologists in the room, and there are probably maybe molecular biologists in the room. And I'm going to talk about psychology and molecular biology, which I do via collaboration. I'm really an electrical engineer by training, roughly speaking. My undergraduate degree is in biomedical engineering from Louisiana Tech. I was a very early student in their program back in the very early 80s. And my PhD is in biomedical engineering from Johns Hopkins. And I've been in this field for decades now. So I'm a department chair. Department chairs always do this, right? I'm going to brag about my department just for one slide, but this is really for the students. We are a big department. We got 42 tenure track faculty, 51 primary faculty, big department, lots and lots of students, and 5 departmentally based training grants, 4 of which are NIH T32s and 1 NSF in our team. We have a number of strengths. We don't try to do well in everything. We try to build these clusters of success. And for any undergraduates, I know there's at least one undergraduate in the room. For any undergraduates who might think about coming to the program, we spend a lot of time on rotations, which have the advantage that they give the students the way to sort of jump from lab to lab during the first year, and then find the lab that really suits the best. Now to my lab. This is the membership of my lab from the last 7 years or so, 8 years. And we're never all in the same room at the same time. People scatter to the wind once they get their degrees. And we'll talk about many of these people in more detail later. I'm wondering where's the printer of this thing? Oh, that's it. And our funding is mainly from NIH. We'll have some NSF funding as well. We've had corporate funding and as well, but it's really largely an NIH funded lab. So the problem that I got interested in when I was living in Ruston, couple hundred miles south of here, was I thought, man, figuring out how the brain works and then doing something about it, what a great problem. And I was right about that, but perhaps naive in my expectation of getting to translational. I imagine we'd be doing, I'd spend the first 2 years of my PhD on learning some stuff, and then the last 2 years on translation, a little bit early. But of course, there's been a ton of explosive activity that's really impacting patients on the translational front throughout my career. We use a number of techniques. I trained as an electrophysiologist. We do a lot of closed loop electrophysiology. So we've built a system for building sort of virtual realities for neurophysiology experiments that we use quite extensively, including in some of the work I'll show you today. We do electrophysiology in behaving animals. And I do that really with the help of psychologists because I don't really, and by the way, our animal model is almost always mice. And with time, we do a lot more optical imaging. And we've always had a strong, as Ryan mentioned, strong computational component to our work. So I'm going to talk about hippocampal formation today. And this is a structure that sort of sits at the top of the hierarchy. It's very far removed. Top of the hierarchy sounds arrogant. It's very far removed from the sensory world. And so the brain builds models of the world and then delivers them to structures like the hippocampus. And the hippocampus has this role of laying down significant events and holding them for a while and then offloading them for permanent storage elsewhere. And Radu mentioned this, when you have, most sleep is so important. I struggle to get enough of it. I'll bet many of you do too. I probably drink too much coffee for one thing, but it's incredibly important for the function of memory. And you can kind of sense that, right? You study for an exam, you wake up kind of knowing the material better than you did when you went to bed. And that's sort of the hippocampus and neocortical structures working in tandem. So we're going to be spending our time in this set of structures today. And it doesn't matter too much for this talk which structures, but we'll do a lot of optogenetic stimulation in dentate gyrus, and we'll do some optical recording in region CA1. Okay. Hippocampus is very prevalent in humans. It's very deeper. We have much more real estate devoted to neocortex than rats and mice do. I date myself that this was a very impactful movie about functioning of the hippocampus that made Chris Nolan, who's now about to sweep the Oscars, we think, into a big star. All right, so. What we want to do is, again, have this balance of like learning things and then using those things. And one of the challenges that we have in hippocampus is it's a harder problem for us than it is for, say, people studying vision. Now, if there are any vision neuroscientists, I'm not being mean, I'm not trying to be insulting. I'm not saying my problem's harder or better than yours. I'm saying there's a specific reason why it's pretty hard to think about building a prosthesis for memory function. And we know we need better treatments for memory function because the treatments we have for dementias are not very effective and they're extremely expensive. So it's almost the worst of both worlds. But imagine if you wanted to build a visual. We have a person who is blind, but the circuitry from the retina on still works. So you could imagine then that the retina just has a map of 2D space. Each retina has a map of 2D space. And so you could imagine you could just have a camera and you could then have an electrode somehow in some way to electrically stimulate the retina in the right space. And you could just basically move that stream of information from the camera into the brain and it would work pretty well. And there actually are companies doing this. This is from a nonprofit associated with that work in Australia. Now, the problem that we have in hippocampus is that there's not a quote map, meaning that for example, I will remember the turn off Shiloh Drive into this place for a long time because I missed it. And man, the punishment for missing that turn is severe. And it's a hard turn, right? Because you see it, but you don't think, I don't know what that is. And then it's, the opportunity is over. So when I came the second time around, I thought, I'm just going to take every turn every turn I see until I get the right one. But the thing about laying down memories is that the neurons in your hippocampus that are representing the laying down of episodic and other relevant memories, they're not like together because they represent the same experience. So they're sort of scattered throughout the hippocampus and they're connected to each other. And it's kind of mysterious. It's just sort of a constantly rewiring part of the brain that's constantly sort of searching for meaning and reward and salience in the world and laying down the things that really have a lot of salience to you. And so what that means is you can't just stick an electrode in part of the hippocampus and expect that, oh, I can shock this part of the hippocampus and recreate that or boost that memory. It's more complicated than that because the cells are interconnected but spatially distributed and constantly rewiring. And these are data for one of my closest collaborator at this moment in time, Steve Ramirez, who's an extraordinarily successful molecular biologist and psychologist at BU, and a student that we worked with together, Monica Chipote. And what we saw in this using molecular tagging events, we sort of looked at the spatial overlap of positive events, positive memories, like for example, word sugar dividends or whatever, and negative memories like foot shock, static electricity shock. And we saw that they're partially spatially segregated, but not well spatially segregated. Excuse me. And they're just all a image. So this really makes this makes that memory boosting device pretty hard thing to think about. And we'll get back to that at the very end. Okay, there are some a couple of things I want you to know that are going to be important for our work. One is that there's a lot of spatially and temporally correlated activity in hippocampus and in a lot of parts of the brain. And you pick this up with like nice periodic looking EEG patterns like this. And we there are two such rhythms. The fast wiggles you see here are called the gamma rhythm, and the slow rhythm, which is about 5 to 12 hertz, is the theta rhythm. And you can see I've taken here is taken this trace and I've the low pass filtered it so that we can see the underlying quite regular activity. This is not our work, it's the work of another colleague of ours from BU. It turns out you can look at the relationship between the theta rhythm, the slow one, and the gamma rhythm, the fast one. And as animals, in this case, this was a rat, it's also true in humans. I think I'm fairly sure it's true in humans. It's been shown to be true in humans as well, that the interaction between these gets stronger as the animal learns something. And it's not, the details don't matter, but it's essentially the details of course matter, but they don't matter today. But the basic idea is it's called the phase 0 through 360 of the theta rhythm is predictive of amplitude of the fast rhythm. We'll get back to this in a few slides. All right. Another friend of mine, one of the reasons I love working at BU is that we've got this amazing community of psychologists, engineers, molecular biologists, MDs who are all working on problems together. Another good friend of mine, close collaborator of mine, Mike Hasselman, had this idea several years ago now that it's still holding water, we think, which is that one way that the theta rhythm is functioning in hippocampus is that the hippocampus is switching between laying down new information and spitting out already learned information in each theta cycle. So as engineers, we might call this multiplexing, phase multiplexing. So at some phases of theta, here at the peak, the system he hypothesized was encoding information, and 180 degrees away, that's tens of milliseconds later, it's retrieving. So it's a constant device that's sort of both learning and telling. It's learning from the rest of the brain and telling the rest of the brain what it thinks is going on. And finally, one last technical setup. I mentioned Steve before. What Steve is really good at, he in his PhD work, which he's still pursuing very aggressively as a professor, he found ways to molecularly tag the cells that had been involved in a given memory. And the way that he believes that the way he does this is that he looks for the levels of CFOS activity. How many people here know what CFOS is? Yeah. So CFOS is a measure of like how active neurons have been. It's a little messenger that the cells that ramps up in its expression when cells are very active. So what Steve did and does is that he finds ways to basically molecularly look for the cells that are really active during a short time window of say minutes or hours, and tag them. And what I mean by tag is make them express a new protein that could make them electrically excitable, light excitable, optogenetically excitable, or make them glow in the dark, make them fluoresce, anything like that. So it's incredibly cool technology. It's honestly, it's surprising that it works, but it does work. And we've done, he and his lab and my lab, because we have to do this to publish the work. We do tons and tons of controls to make sure that this is what's really happening. And it really does appear that this is happening. So we're going to use this technology to think about this hypothesis, this computational model. So the first story I'm going to tell you was led by a former student of mine, Bahar Rasapar, who's now off in biotech and having a great life. But she decided, based on our discussions and reading these papers, she decided, well, what we need to do is basically stimulate these tag cells while measuring the local field potential, while measuring the EEG, the little micro version of the EEG, and stimulate. If we can stimulate selectively at this phase, then we should basically be working to encode. And if we stimulate on this phase, we should be driving a recall event. So the idea then was that if we train an animal, in this case, we're going to foot shock it, so we're going to fear condition it. We train the animal to be worried when it's in a given environment or has a given set of neurons firing. We should be able to get a stronger response, stronger fear conditioning when we stimulate at the trough. So to do that, we've got to basically do a lot of real-time computing, and that's where this is the one thing the engineers are like teams like mine are really good at. We could build like really high-speed systems that go along we measure this local field potential in real time. And we predict when's the next peak, when's the next trough. And we can do that with quite a bit of accuracy. It's harder than it sounds because there's a bunch of noise and this is not a perfectly periodic signal, but it's doable to plus or minus 10%. Okay, so the setup looks like this. We're recording the EEG. We filter it. We get a nice look at the theta rhythm. We predict the next peaks in this case, or troughs in other cases, and then we at the right time stimulate using a laser the tag cells, which are tagged to be optogenetic. So you shine, in this case, you shine a bluish green light on them, and that makes them fire action potentials. And our idea is that's going to drive the memory, and it's going to depend on the details of whether we stimulate the peak or the trough. Sounds easy, right? Okay. Yeah. For hard effective effort, I'm sure it was not that easy. So I'll go through our protocol. It's not. I just want the people who think about these kinds of experiments to understand the protocol. I'm not going to do it in much detail. But we take an animal, we get it used to interacting with humans. And we put it in environments where it's basically perfectly happy to wander around and sleep around and see what's going on. On day 5, we put it in a mode where we can tag cells. We take it off doxycycline, which allows us to tag such cells. And we put it in a new environment that's indicated by this cube rather than this triangle, and we give it a foot shock. So it's like a static electricity shock, and it doesn't hurt. It doesn't damage tissue. It doesn't hurt them. But boy, they hate it. And I got to say, I hate it too, right? When you touch a doorknob and you get your fingertip shot, it's surprisingly unpleasant, even though it doesn't hurt you. And the rats seem to feel this, the mice, sorry, seem to feel the same way. Then we put them, later on the next day, we put them back in that box, and sure enough, we see that they are like, I don't like this place. This is the place where I got that static shock. And the way we tell that is that they're instead of wandering around happily looking for things, looking to see what's going on, they kind of sit there. They're much more prone to they freeze, which is what mice do when they're prey. Finally, we put them in the happy context, the neutral context, and we reactivate the memory. We try to reactivate the memory, and we see how much does the animal freeze. And we do it for a bunch of things. Like I said, we had a bunch of controls in this paper, which just came out last year in Eli. But I'm going to focus today on stimulating at the peak and stimulating at the trough. So I'm going to stop for a second because it's kind of a complicated story, but I've told you the most complicated parts, and I think it's going to get easier now if you pretty much follow what I'm saying. So any questions at this moment? Yes.
Stimulate the peak and the trough. Choose.
We choose one or the other. That's right. Good question. And we also just do the data I'm not going to show you, or we just stimulate with a clock, bang, 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 and we use that as another control. Yeah, and we have controls. We have other controls, animals that we did not foot shock, right? They go through the entire experience, except they never got the foot shock, and they'll of course not freeze when we put them in this environment, and they won't freeze when we hypothesize they won't freeze when we operate. Okay, so let's take a look at the data. After all this work, I'm glad Bahar's not in the room because she would think, like, I worked for 5.5 years on this, and you're going to basically show one data slide. I'm going to show two or three data slides. But this is for, the most important one is, so we're looking at the amount of time that the animal froze, the proportion of time that the animal froze. And this is a paired comparison, and you can use any stats you want. We put non-parametric stats on this with a non-parametric paired test, and it's a strong effect. It's well below p equals 0.01. So it looks like it works to the degree. Now you do get some freezing when you stimulate at the peak, which is the wrong time to stimulate, wrong phase to stimulate, but you get more freezing when you stimulate at the trough. I'll show you another part of this. I'm cheated a little bit because I told you about this thing that we call MI, which is the relationship between theta and the relationship between theta and gamma, the slow rhythm theta and the fast rhythm gamma. And they're more connected when animals know something about the world, like have been trained in something. And what we saw is, sure enough, when we do trough stimulation, but not peak stimulation, we increase this measure. So we increase the amount of theta gamma coupling. And we can also look at the animal's behavior and say, okay, we know on a given trial how much. I think one of the reviewers has to do this. I mean, it's a good idea on the reviewer's part. We had really good. I mean, all reviewers are ferocious, right? But some of them are ferocious and help you. And we got lucky. We got lucky. These reviewers were ferocious and helped us. So they wanted us to take a look at this, and sure enough, it works. For the trough stimulation, you get the relationship you might expect. When the animal freezes, on trials in which the animal froze effectively, meaning that the memory really seemed to hold, the theta gamma coupling was higher. So there's a fairly clear relationship between the amount of freezing and this theta gamma ratio. That was not true for peak stimulation. So what we take to what we speculate, there's always the speculation part of any paper. What we speculate about is that we think that when we get freezing in this case, it's sort of a recollection of memory, but it's less natural in the sense that it's not accompanied by an increase in theta gamma coupling. Okay. Another person joined the team, Jacob Norman. Jacob just defended. He's writing up his papers as I am here. And Jacob started by, we wanted to start looking at ways to do recordings of lots of cells simultaneously. So the ideal would be, there are a few ways you could do that. You could put a bunch of electrodes in the brain, you could image, or you could image. So we're going to choose to image. And in this case, we're working towards doing imaging of membrane potential, but in this case, we're imaging the amount of calcium in the cells, which is a very strong correlate of the level of activity. And I'll just show you, if this works, I'll show you just what the raw data look like. So, you know, this is we're using a 2-photon microscope. We have about, 50 to 150 cells in the field of view. And we can see them when they light up and when they don't. And every once in a while, a cell just is always lit, which means it's dead, something pathological is going on. And we can pull out these traces. They look like this. And we use a molecular marker in after the fact analysis to determine which cells were excitatory and which cells were inhibitory. And that's you know, there are a few ways to do that, but the way we did it was just to use a virus to basically have another fluorescent compound at a different color in the inhibitory interneurons. So Jacob wanted to sort of take this to the next level, and what drove him, you know, behind every student, there's this idea that they're obsessed with this, every PhD student, and his idea was, I want to see what happens when the animal freezes versus when it doesn't. Like, what's going on? Can we distinguish between the activity in a downstream structure that that represents this memory, and I'll know it if I see this pattern of activity when the animal freezes and not when it doesn't. Okay, so a little bit. This was a complicated experiment, and Jacob probably regrets his ambition, but he did make it out in a little over 5 years, so it's not too bad. What he wanted to do now is, this is the dentate gyrus. This is the structure where we've been optogenetically. So we're doing the tagging the same way. We again fear conditioning animals, and we tag those cells to basically that represent that memory in this part of the brain. In another structure, CA1, we're also tagging engram cells. So we're also tagging cells that represent it. So we have two tags, two populations of memory encoding or engram cells tagged. One in the input structure, one in the output structure. The flow of information goes like this. So we tag these and we stimulate them with light, and we record up here. And we use the same trick that Jacob used in this paper to record two populations of cells. Only in this case, it's not going to be inhibitory and excitatory cells. They're all going to be excitatory, but it's going to be cells that are engram cells that represent the fear condition, and cells that are not engram cells that represent other experiences of life. Okay, so a couple of things that are really complicated. One is, we don't know how to do this in a freely behaving animal. So we had to, it's possible to do it maybe, but we didn't do it that way. We actually trained the animal to do the same protocol while sitting on a styrofoam ball that is suspended on a column of air, so the animal can kind of run around and be happy. Its head is fixed, and we're looking at its hippocampus using a 2-photon microscope. That took a while, but it did work. The second thing we had to do is we had to worry about the colors that we were using, and we had to basically, instead of using the traditional version of channel rhodopsin, which is excited by blue light, we found one that had just come out in the literature as a red version of channel rhodopsin called Crimson R, and we paid someone in North Carolina to build a virus for us so that we could use it instead. So the idea then is that we're going to stimulate with red light and record with green light. And we're going to do it at multiple depths, but we're going to look at this part with very high resolution and look and see what the cells are doing. So I'm going to show you, we're doing okay. I think we're doing okay on time. I'm going to just show you a little bit of Jacob's data. That's interesting. You're the first people outside Boston University to see these data. So I better do a good job of explaining them. This is supposed to be a green blob. I don't know why this one shows up and this one doesn't. You know, that's a weird little projector thing. But what we're looking at is the tag cells, the ones that represent the memory in the output structure, and the untagged cells. They represent other things, other memories, other events. And what we're going to do is we're going to ask the question: do we see a difference in when we're light stimulating versus when we're not light stimulating? And the answer in the tag cells is no, we don't. We see the same range of estimated firing rates in both tagged and untagged cases. So the cells that are representing memory, they're doing the same thing, whether whether we're stimulating or stimulating the upstream fear memory or not. So that surprised us a lot. We thought, well, these cells represent that memory. Of course, they're going to be more active when that memory is being induced. That artificial memory is being induced, but that's not the way it happened. Another thing that surprised us and kind of saved the story was that the untagged cells, which represent other memories, they actually have this doesn't show it super well, especially because of the projector issue, the display issue, but the rate actually drops by about 20% on median and on average in the untagged cells. So instead of the cells that encode the memory going up in their rate, their neighbors that encode other memories drop their rate. So I did not expect that at all, but the data very clearly show that. And that's a highly significant result. So we did the statistics very, very conservatively. Didn't make any assumptions in these stats, and this result still came out as an extremely significant result. It was significant in every animal, and it was very significant, highly significant in the some data processing.
So John, if just to confirm, when you are stimulating the fear cells, then you are recording what is the response of the output cells. And if they were conditioned, it didn't matter whether you stimulated or didn't matter, but the neighboring cells that were unconditioned, when you induce the fear memory, it got inhibited.
That's right, they get inhibited. And we don't know, so obviously not one of the next things we're going to do is try to find the circuits that inhibit the other cells.
Other ones. So
Right.
something else is getting activated
That's
that is
right.
causing that
That's right.
inhibition.
So I think of it, because I think it's like a signal to noise issue. What you do is you just shut the rest down. And there's a lot of examples in neuroscience, in neurophysiology of this. Like it's not how fast the cell is firing that represents your fingertip when you touch your fingertip on something. It's not only that rate goes up, it's also that the rates that represent other parts of the finger might drop. Right. So somehow these cells, the circuits that drive the memory cells, are somehow inhibiting their neighbors to give you better signal to noise. And I was a little bit worried that I would get caught up in complicated graphs and drive you folks crazy. So I decided to text my way through this part, but I promise you everything I'm saying is we firmly believe. And we'll put this on. We've really become big fans of bioarchive and those kinds of things because I like sort of getting things out there, finding out what my friends and colleagues think about the work. So we'll be posting this paper on bioarchive in the next month, I'm sure. I hope. Almost. And so what I didn't show you is that you remember the non-tag cells, as Ronnie points out, the non-tag cells get suppressed when you evoke the artificial memory. And this is in the output layer. We're stimulating the input layer, we're recording in the output layer. Similarly, we can look at when the animal responded, when it froze or stopped licking in this case, and when it didn't. And we can look on a trial by trial basis. Is there a difference? And there is, but only for the non-tag cells. So it's completely consistent with that other result that if I try to evoke the artificial memory once and it doesn't work, then I see control behavior. And if I stimulate and I evoke the memory and the animal the animal does what it does, it freezes or stops licking, then it shuts down the competing cells. So very consistent. Finally, this result fades away over the course of several days. So we did these experiments for several days in a row, and eventually the animal sort of figures out, like, you know what? When I have this memory, nothing bad happens. And let's see, is this extinction? It's extinction, I think. I struggle with the difference between extinction and habituation because I'm not a psychologist. But we extinguish the memory over the course of several days. As we extinguish the memory, these differences in firing rates, they just go away, and we're back to where we started. So, like, it's a fresh slate as far as hippocampus is concerned. Okay. So I think I've already told you all these things. Another look at the team. This work was mainly driven by Bahar Razafar and Jacob Norman. Jad Noyad that helped with a lot of signal processing. 2 undergrads, Melanie Quick, who's now a med student at Minnesota, and Ben Lahner, who's a double E student at MIT, who did a lot of the algorithm and programming for us. And Fernando Fernandez was, until very recently, my right-hand man. When he moved to NIH, he's a program officer for the Brain Initiative now. And which is great for him, and good for you, but not so good for me, because he's in conflict. He can't handle my guts, which is fair. Okay, so I wanted to just talk a little bit about where the next things are going, and then I'm going to get back to this kind of more general question, which I think is interesting, in particular to this audience. You know, we are looking at more detailed analyses, and I've got a guy in my lab, Danny Carbonero, who's looking at these because these are really rich, gargantuan data sets, and Danny is really good at advanced data analytics and machine learning. He's diving into these data as we speak. This is going to come up again. You know, there's this fight between the cells that represent the memory and the cells that don't. And we wondered, like, can we, in a less detailed, biophysically detailed manner, improve the function of the brain? And we'll get to some work by other labs that has tried to do that and seemingly has worked. And you know, Akemi Ito is a new student in my lab. She's looking at stability of representations over time. An animal has a memory, and we optically record the neural correlates of that memory. How do they change over days? We probably can't go out to weeks, but how do they change over, say, 1 to 2 weeks? Okay, so I am going to end by just talking a little bit about this problem of, can we make a prosthetic for episodic memory? Huge problem. I think we all know your probability, sadly, your probability of really pretty significant dementia grows very, very fast once you turn about 85. Even if you're in good health, it matters how good your health is. And I'll just say, the number one thing to do is, these are all things that I struggle to do myself. So I'm reminding myself, like, exercise a lot, especially aerobic exercise, eat well, especially vegetables and blueberries and things like that. Not so much meat, don't smoke, don't drink. And I, of course, had I drank with a couple of you last night. So okay, so you could argue from results like mine, you could argue, like, look, you can't really make a prosthetic for episodic memory in hippocampus because there's not a map. You can't just stimulate in this part and recall that memory. That's the pit. Now you could imagine maybe one day we'll be able to have like 20 different optogenetic compounds that we could load 20 different memories with, and we could select, I want memory number 18 and turn that one on. But you know, that doesn't seem like a productive strategy. And so I didn't want to end pessimistically, because I'm not pessimistic that we'll be able to do things. I mean, there are plenty of things that are going to be done. I'm going to focus on the 2 device related ones. And the first one is from a group at USC. So and Wake Forest. The neuroscientists are at Wake Forest. The engineers are at the University of Southern California. And Ted Berger, whom some of you may know, led this team, and they had an NSF-ERC about this. So they actually just kind of built this kind of pretty simple model, multi-input, multi-output model. It had maybe 20 channels. And they tested it recently, relatively recently, in human subjects. So why were they able to do that? That sounds unethical, but of course, that's not the reason that people were implanted. People were implanted because they have intractable epilepsy, and they go into the epilepsy clinic. They get electrodes put in their brains, several electrodes put in their brains, because the epileptologists are trying to find the source of the seizure. And the hope is that it's in a spot that they can excise surgically without causing significant neurological damage. So that's the real reason these patients are implanted. But while they're sitting around bored, they can agree to be participants in an experiment. So Ted's team basically took several of these patients, and they took this really simple device that basically just records some activity and then stimulates in another spot. You know, it records on some electrodes, some of these electrodes, and stimulates in other electrodes, and this is really simple model. And you know, and then they did a delayed match to sample, which is a standard psychological test. It's hippocampal independent, and you know, they got an improvement in performance. And honestly, I've known Ted forever, and when he first told me this, I thought, Ted, this isn't going to work. And he thought, he was nice about it, but he thought, oh, ye of little faith, right? We're going to try it, and we're going to do it safely, and you know, it did work. So I think what they're doing is not significant, not exactly representing memories, but they're stimulating in one spot versus another spot, and it seems to help the performance. So good for them. Now there's an even less invasive. Now, you have to have electrodes, or you have to do this, you have to have electrodes or some other way to stimulate the brain. So we don't, you know, Elon Musk thinks we're all going to be walking around with 10,000 electrodes in our heads in a decade. I'm less convinced. I would not sign up for that under any circumstances I can imagine. But you know, and again, they only did this because the patients had the electrodes in their brains for other reasons. But this is a device that is, you know, it's a little controversial, but you know, there are some papers out that suggest that you can do other things, just kind of juice the brain a little bit. And so this is from the Weill Cornell website. There's several groups working on this. And what they do is they just put some electrodes on your head, and they pass DC current through your brain. And what it does is it on the anodic side, it makes the extracellular space a little bit more negative, which makes the cells a little bit more excitable. And so it's a way to just kind of juice activity a little bit. Seems safe. There's a great deal of controversy about whether it's effective. It's a pretty new technology, but I don't really think there's much danger in it. You know, you're just passing a little bit of current through the brain. Not too bad. And you know, they claim that these folks claim that they are getting improvements in memory performance. Now, you know, big pharma has been working on drugs for epilepsy. I'm sorry, for memory disorder, for Alzheimer's and other memory disorders for a long time, and they've mostly been huge flops. There are some drugs, like you can inhibit the enzyme that degrades acetylcholine, and that helps a little bit, but not a ton. There was one antibody that was approved and then pulled off the market, and it was extremely controversial because their results were like, and the FDA honestly, because I think they feel like it's such a big problem, we might as well try. They approved it. There's a new antibody-based one that looks much more promising. So maybe we're going to get there. We have to do something, right? And with that, I'm going to wrap up. As I've already told you, my work is funded mainly by the NIH and a little bit by NSF. And I just wanted to point out two really important collaborators: Steve Ramirez, who's an assistant professor, soon to be president of the world in psychology and brain sciences at BU, and Mike Cassimo, an old friend of mine, who is the person that developed the model in the first place. And I'll end by showing you a little. This is my building. This is my street. This campus does not look like the University of Arkansas. It's a very much an urban campus, and I'm on the 8th floor. I'm sitting in this floor of this floor. And I'm happy to answer any questions you might have. Yeah.
I'm just curious, when you were evaluating ways to tag, has anyone tried?
No, not that I'm aware of.
John, if you could repeat the question.
Yes, sure. The question was, has anyone tried tagging with nanoparticles? So presumably the idea that you could get more selectivity, perhaps. You could tag with nanoparticles that you could then use to excite the brain, maybe through heat or other means. To my knowledge, nobody's done that. You'd have to, I think what you'd have to do is you'd have to, I don't think you can encode the nanoparticle necessarily genetically, could you?
Right, and you can't say we can give each one a field.
Yeah, it's a pretty interesting idea, and I think I like where you're headed, right? You could imagine that you could have a lot more addressing and get at the specificity problem, which is the real problem that we have.
And have you bought a quantum computer yet?
I have not bought a quantum computer. We have a lot of people really interested in quantum computing at VU, but I have not bought one yet.
I think you'll need some pretty soon.
Dr. White, we have a question online, a couple of questions online from viewers who are watching the presentation. And if you could just summarize the question also
Sure.
for our Zoom audience. First question, do you think that there might be peripheral pathways that might provide generalized activation, juicing, of these hippocampal circuits, similar to the non-invasive cortical stimulation, but activating a peripheral input?
Right. So the question was, are there other pathways that we could perhaps stimulate that drive juice the hippocampus and make it more active? Which I think, that the most used class of drugs operate somewhat on that principle. So they're cholinesterase inhibitors. So they basically meet they have the effect that the acetylcholine that's in the structure, which tends to excite neurons, sticks around longer. And what they see in Alzheimer's patients is that they get about 6 more months of good performance, and then it drops off. So it leads to a delay in the presence of symptoms. And look, if you have Alzheimer's, then 6 more months is great, but it doesn't last. And I would suspect that that's because the damage gets so much worse over the course of the next 6 months. This is speculation, but that's what I suspect is going on, and that might also affect us. So it's a really good question. So you could stimulate, you could probably stimulate either cholinergic pathways, or you could stimulate maybe noradrenergic pathways, and they certainly innovate the campus. So I like the idea.
Another question online, and again, if you could just summarize that before you respond. So have you looked into tagging hippocampal cells engaged in ripple or spindle generation, or is that what you mean by engram cells?
Have I looked at tagging cells that are involved in ripple or spindle generation, or is that what I mean? That's not what we mean. We mean cells that were active in a state that is not like the ripple or spindle case. So we could do that. In theory, we could do that. I'm trying to think about the challenge that we have. So ripples and spindles, they appear in other states that are like states like grooming and in some sleep states. And so you could, and so if you want, it would be very interesting to tag the cells that are involved in those states and then drive them and see what happens. That's a cool experiment. The challenge would be that the ripples and spindles are short-lived, and the windows that we have for the tagging are long. So we take the animal off doxycycline, it clears out the doxycycline, and then we can tag. And that's hours, not seconds or milliseconds. So we'd have to come up with a better way to tag. But I think in principle, we want faster ways to tag cells, like in very short time windows. That would really be helpful to us. We don't yet have those, but if we did, we could use them for an experiment like that. Yes.
Again, thank you, John, for this stimulating talk. So I had a question regarding your overall experiment plan, so if I can specifically mention that it's based on fear conditioning. Fear memory. Just a very basic question. But if you were to do the opposite, I don't know if that's possible or not. That's like a confidence conditioning.
Yeah, confidence conditioning. I love it.
What would happen in these times?
Yeah,
And
yeah.
it's briefly-
Yeah, absolutely. So the question was, the question was, it's a good question because if you can only do fear conditioning, then, and you can't ask the animal, are you afraid, right? So you have to look at it. Does it stop licking? Does it stop moving and infer that it's afraid? And it sure would be nice if you could do the opposite kind of conditioning, which Raj called confidence conditioning. I like that term. And yes, you can do that. You can expose an animal to, there are a few ways that that's been done. You can expose an animal to something really delicious, and they will remember that, you know, if these are animals that, you know, they get a hold of something like, you know, Fruit Loops or something like that. They love Fruit Loops. So then you can then label the cells that associated with that happy time and drive them, and what you'll see is the animal gets more active. And so that it's basically activity, the animal's physical activity. The happier it is, the more afraid it is, the less it really goes into this crouched. When it's afraid, it goes into this crouched crouched freezing. And when it is happy, it's just running around more. So the proportion of time that it's freezing, the speed at which it's running, these are all indicators of its level of vigor and happiness and confidence. Yes.
Then what about this question in terms of encoding memory? Can you comment on whether there were whether all the neurons that were encoding a particular memory are equally important, or whether some of them are more important than others? Or in other words, we have to stimulate all the neurons to involve to another thing, some of them.
Yes. Yes. The question was, do you have to stimulate the entire set of, let's say, 50 or 100 neurons to evoke the memory, or could you evoke a small fraction of them and get that and get the same behavior? So that's a really interesting question. And I believe the answer is, if you can do that experiment, you should be able to evoke the memory, because there's a lot of, you know, there's a kind of the classic theory of hippocampal function is that this part of the brain where we're doing the stimulating I don't think you need to have a perfect full representation, that there's a downstream part of the brain that will do the pattern recognition. So there's a part of the brain between PG and CA1 called CA3, which tends to do pattern recognition. So I think if you had half the pattern, you probably get less effective recall, but you would still get recall. Yeah. But it's a challenging experiment to do, because we're not doing selective stimulation. We're just shining a laser in there, an unfocused laser in there, driving all the cells that it hits. But you could, if it's possible to do a little focused stimulation with that laser. And if you could do that, we can start to answer that question.
And when you stimulate, what's the pattern of the effect that they how the 100 stimulation? You said you had a frequency control, but
Yeah.
how do you get?
Yeah, the question was, what's the pattern of the optical stimulation? And it's a good question I didn't cover. It's several brief pulses. The pulses are, I think, 10 milliseconds, and set in typically groups of 5, and every 100 to 200 milliseconds. The pattern of the stimulation, as long as it's sufficient to drive the cells, it doesn't seem to matter very much. We actually looked at that a lot in an in vitro prep where we had much more control, and we could do patch clamping and other things to see it. It doesn't seem to matter so much. It tends to just we're just trying to generate enough spikes to kind of represent the memory. Yes.
So with the emerging organoids right now, so opportunities to gain.
Yeah.
Any
I'm super excited about organoids. I don't know how to, I personally don't work on them, but I just think, in the end, we all know this, that mice are great animal models. They are not humans. And when the closer you get to the disease, the more problematic that issue becomes. There's this, kind of classic thing that we've cured cancer many times in mice. And so what I think is very exciting about organoids is if you could build human-derived organoids that represented this kind of structure well, I mean, it would be interesting. Would you have ethical issues? I don't know, but if you could do that, then I think it would be fantastic for translational purposes. It's, it would mean less animal use, which is always a positive, and it would get us closer to to applying things in humans because we can use human-derived cells. Organoids seem like this is gonna be the century of the organoid, right? And I'm sure that's true in brain too. So there is a group of folks at BU working on organoid cultures for neuroscience research. You have to reconstruct a lot of development to get it, work. It's hard, but
So
yeah.
one comment and then one question and then a comment. So you showed Ted Berger's stuff at the end, where it was basically what you said, just kind of conditioning, overall diffuse conditioning of the brain and changing its state, right? Okay, have you examined that kind of a situation in this model itself, where all you do is just change the state? And, if you go back to your dynamical systems theories, then if you just bring them, to a boundary, then you could easily trigger.
Yeah.
the other.
Yeah, did the audience hear that question? Yeah.
No.
Okay, so that's a great question. I love that question. We haven't done that. We were just trying to get it to work.
Mhm.
But I love the idea of saying, like, okay, now that we've satisfied some of our curiosity, right? We know that there's something in the Ted Berger work. And so could we start to think about ways to improve that approach? And that's a really good idea. So, one of the things I vaguely mentioned, but didn't really talk about was I think this is it. Oh, how can appropriately phased, quote, tone improve compromised recall? And that is exactly, that's what I was thinking about when I wrote that bullet point. I like this idea that we can do same experiment, but basically say the goal is not to recreate an artificial memory, a precise artificial memory. Instead, the goal is to just drive the circuit a little bit.
Mm-hmm.
And we could do that. And actually, diffuse optics is a good method to do that. And that is something you could do in a human fairly soon, right? There are genetic therapies approved now, right? So you could imagine, it's not now, but you could imagine in our lifetimes, in my lifetime, and I'm an old dude, that you could basically drive a little bit of tone in cells that tend to drive good memory performance by using optogenetic methods.
And then if that happens, then maybe some other, something in the environment triggers your memory, and that is enough to cue your brain to remember the memory.
That's right. That's right.
So-
That's right. And you could argue that, if that can get, if you can get a person who is in their 80s or has memory deficits that may or may not be age related, if you can give them just a few more years, that's a huge benefit to them, especially if you can do it in a way that is not that expensive. And you're right. I love this idea of just don't worry about the specificity. Just try to tickle the network and get it more effective. Yeah.
So a wonderful talk. But I also want to point out, you brought out the organoid story. So I was privileged this last year
to work on a working group for the NIH director for looking at what is called novel alternative methods. And basically, it is a lot of it is about the organoid stuff. So if you, we are very, very satisfied and very happy that the working group's recommendations and the report was taken up by the ACD and the NIH directorate. And just last week, I think I sent out an email to people around here. There is a whole basically an announcement from the new NIH director of this whole area that is going to get further invest. Basically they're going to be looking at it and there'll be further investment. So I encourage you and others to look into this recommendations and read the report of the working group of what all we have recommended. And it is a very comprehensive recommendation from everything from training that is needed, and besides just not just talking about, okay, research, but what will be needed to actually get this technology to be standardized, not just in labs, because if this is really going to be something that becomes a model system to be used, you need much more than multiple labs saying, oh yeah, I can do an organ and I can do this, but you need, so there's all of that, including also the training processes, how you're going to train the next generation of students, researchers, and then think about really a very fast forward future of maybe where you might even have devices and organoids linked together.
Yeah.
And so, the future is amazing.
Yeah, it could be, that would be so exciting. It would be so many advantages. You're right. Standardization is important so that pharma and biotech could be using the same organoid systems, maybe at a larger scale than the labs are using.
And device manufacturers. So I think right now, most of our device manufacturing in particular, pharma still has got some things they're using, but our device manufacturing is very much a trial and error thing,
right?
And so this working group also talked about when it's a novel alternative methods, computational methods, digital twins, that all in silico, in vitro, in chemical combination, but then getting all of these together. So that's why I love what, you've already you've been a pioneer in a way of doing this merging of things, right? In the basic science part, but then in the device manufacturing part, how can we use all of this to actually change the way in which we actually make devices that might go into people, or that are designed to go into people. So using this model system in combination with computational stuff and moving forward. Any other questions from anybody? If not, let's give a big round of applause. We are going to have time with Dr. White now. Some will go to the lab, but I think now first thing is students.
Yes.
And so I think your opportunity to obviously ask Dr. White technical things, but more importantly, I think being able to ask him, what does it mean for going through research, what paths to take, whatever other questions that you might want to ask. And so undergrads, grads, everybody, you're welcome to, this is the time we have set aside to be able to talk with you all. And all questions are, how do you choose, if you're an undergrad, how do you choose a lab to go in, an advisor to go in? How do you manage communications? It's a wealth of knowledge, wealth of guidance, and so it's an opportunity for you all to talk.